In this video, I'm going to give you kind of a primer on plant physiology. And by that, I just mean I want to give you the kind of fundamental physiological processes that you'll need to understand in order to be successful in this class. So I realize some of you maybe had plant biology or plant physiology and have been introduced to those concepts, but I know some of you haven't. And so this will get us all on the same page and will help you kind of see how I think about the world a little bit more. Okay, so what we're going to cover in the plant physiology primer are kind of these four topics. I'm going to introduce you to the idea of carbon budgets and how kind of plant ecophysiologists like myself think about the world of ecology and the world of plants. Then we'll cover the different types of photosynthesis and think a little bit about the trade-offs of those different types of photosynthesis. So that we can start thinking about how the type of photosynthesis a plant might do could impact its distribution on a landscape. Then, and those two things will be in this video, and then I'll have a second part that will kind of be for next week, where we'll talk about the nutrient demand of plants and nutrient uptake and nutrient use, and then we'll talk about water use in plants. Okay, so let's get started with thinking about carbon budget. So if we think about the goal of plants, in ecology, we generally think of fitness, right? The ability to reproduce and kind of spread your genetic material or your genetic offspring across the landscape. That's our ultimate goal of organisms as we think about it in ecology. A pretty good corollary for reproduction is actually growth rate, or at least growth rate in a particular environment. So in general, the plants that grow the best in a particular environment are also the ones that reproduce the best. Not 100% of the time, but in the vast majority of times that's true. And in the literature, if you look at research articles, a lot of times people will use relative growth rate as a proxy for fitness. And so it does a pretty good job of substituting for that. So we're going to be focusing on growth rate in this class. And we really have to think about the growth rate of a plant in a particular environment, not just who's the fastest under ideal conditions, but who can grow the fastest or what species can grow the fastest under a certain set of conditions. So in order to think about kind of growth rate and track growth rate a little bit more, we're going to focus on individual elements that are chemical elements that are in plants. And so if we think about, so I guess I need to explain this picture. I need to either take it out or something, but I've left it in here for years. One year when we were in the Arboretum for class, we saw a chicken nugget or a chicken tender on one of the fir trees, and we thought that was kind of funny. So I took a picture and I made some joke about it in class. The joke was really bad and I don't even remember it. So the picture's still there, but not important. I think the joke had something to do with like chicken tenders not being an element in a plant, which is pretty bad. But if we're actually thinking about what elements are in a plant, this is a pretty good list of the most common ones. And certainly we could make a longer list. But there's lots of oxygen, hydrogen, and carbon in plant material in particular. And those are the three compounds that make up simple sugars or sugars in general. So these are just three different kind of types of sugar molecules. This is a really simple monosaccharide. So there's just one of these rings in glucose. If there's two rings, it's sucrose. And then this is when it gets longer and you start linking those chains together. The point of this showing you this, though, is that if you remember back to your chemistry class, at each corner here, unless indicated otherwise, there's a carbon atom. So we've got several carbon atoms here, there's a carbon up here, and then the rest of it is oxygen and hydrogen. And simple sugars are kind of the building block for plant material in plants. So it's what plants produce first during photosynthesis, and it's what they use for energy, and it's what they use to build the bulk of their biomass. And so when we look at a plant, much of what we're seeing is really these three elements combined in different combinations. Um, then, of course, we know plants need lots of nitrogen, so it's another major component. Then there's phosphorus, potassium, and other micronutrients. In general, these micronutrients from phosphorus on down are in abundant supply for plants. There's certain situations, certain places where they are not abundant, but in general, they're not limiting plant growth. Nitrogen certainly can limit plant growth, but it's not part of biomass. And so what we're really focusing on now in growth rate and biomass is what are the major components of biomass. So that leaves us with carbon, hydrogen, oxygen. Now, hydrogen and oxygen, you know, are the elements that are in a water molecule, and we know water is limiting to plant growth. However, like the bulk of water goes towards transpiration, not toward biomass. It turns out getting oxygen and hydrogen for biomass is actually quite simple for a plant. It's not generally a water limited, or those elements aren't limiting to biomass growth. What is limiting to plant growth is carbon. Carbon is relatively hard for plants to get. They have to put a lot of energy into getting it, and it doesn't come easily for a plant. And that gives us a really nice element to start tracking kind of resources in a plant. If we check oxygen and hydrogen, it doesn't tell us that much because we know it's not limiting, but because carbon's limiting and it's part of every single component of plants, it's a really useful element to start understanding how plants grow in their different strategies. So, this is really about the same slide we just saw, but I've added another compound down here, which is cellulose. So I showed you these simple sugars. Cellulose is really just made up of these microfibrils that are made up of chains of simple sugars. So each of these rings is like one of these simple sugars. So it's just linked together. And cellulose that's part of every cell wall in a plant. And so that's really, really common product for biomass. So I just show you that to reinforce this idea that carbon is everywhere and really important for plant growth. Okay, so that is all to try to convince you that carbon is really important and it's something we can use to track kind of growth and plant growth strategies. So let's talk a little bit about the acquisition of carbon, and we'll start talking about why it's kind of a difficult thing for plants to get. So carbon acquisition, as you know, occurs in photosynthesis. So you're all familiar with photosynthesis. I'm going to break photosynthesis down into three main steps for this class. And so you're probably familiar with these steps. You probably know more about them than what I'm going to give you here, but I just want to get us on the same page about what three steps I want to break photosynthesis down into. So the first step is going to be the harvesting of solar energy. So plants take the energy that it receives from the sun and they're able to convert that into chemical energy. And that chemical energy then can be stored and power other functions in the plant. So one of the main functions that that energy funds or powers is the fixation of carbon. So the second step is where plants use that stored energy to grab a CO2 molecule and take that carbon out of that CO2 molecule. And so we call this fixing or assimilating carbon in this step. And then this third step would be taking that CO2 molecule, or that carbon from that CO2 molecule, putting it into a larger molecule and building simple sugars. So our three steps are harnessing or harvesting solar energy and storing it as chemical energy, using that fixed energy or that stored energy to fix carbon, and then converting that carbon to a simple sugar. So those are our main steps. Let me move my picture here so you can see the full word, converting it to sugar. And just to help you kind of visualize where some of these processes are going on in a plant, this is just a few different cross sections of leaves. These are scanning electron micrographs. And so for some reason I like this. Actually, it's got the writing on it. Let's use this figure. I guess that has text on it too, but let's use this picture in the bottom right. So this is a cross section of the leaf. These kind of circles at the top are the upper surface of the leaf, and each circle is an individual cell that makes up that upper surface of the leaf. Then these kind of elongated portions or cells in the leaf, these are referred to as the palisade parenchyma. Then below that you have, you can see some cells and then some gaps in between, and these are air spaces that allow gases in the leaf to diffuse and move around fairly readily. And then this brown structure here is called the vascular bundle. There is a bundle sheath of cells that are around it. And it's hard to make out in this picture. I'll show you another picture later where maybe we can see these structures a little bit better. But we've got this, the vascular bundle with the bundle sheath around it. Up here, these cells, you can't tell right now, but these are phloem cells where the plant moves around sugars and nutrients and other compounds. And then the cells below it are the xylem, where water, primarily water, is moving throughout the plant. The palisade cells, that's where the majority of the photosynthetic reactions are happening. So these have pigments in them that are able to take energy that passes through these cells from the sun, and it's able to harvest that and convert it to chemical energy. And then the fixation of CO2 in the initial steps of photosynthesis also occur in this palisade parenchyma, okay? And we'll talk about some of those individual steps here in a little bit. Okay, so that just gives us an idea of, or at least I've tried to convince you that carbon is important, and we're going to be focusing on carbon a lot in this class. We've talked about the 3 steps of plants kind of getting that carbon into the plant. And now you kind of can see a little bit, or start to see where some of these reactions take place in the plant. Now, if we go back up here to this, our 3 main reactions, this last step I said is converting the CO2 or the carbon from the CO2 molecule to a simple sugar. Once it's got it in these simple sugars, we kind of think of this plant having this pool of simple sugars that it can do lots of things with, okay? And again, we're tracking the carbon in it. So once these sugars are produced, we think about this pool of carbon that the plants have access to that they can do with what they will. So this is where we get into the carbon budget. This large rectangle here is the pool of carbon that plants have access to. So that's like that pool of sugars it produces from photosynthesis. And we can think about carbon budgets on a range of time scales. We can think about it on a daily time scale, a seasonal time scale, an annual time scale, a lifetime scale. So it can depend on what we're interested in. But for me, as I think through this and I talk through this, I'll be thinking a little bit more towards like the seasonal or annual time scale. So let's say a plant can accumulate this much carbon. We won't put a number on it, but it's got this large pool of carbon it can accumulate over the course of the year. And then it can do lots of different things with this carbon. And so these rectangles below it represent the different things or structures a plant could build from this pool of carbon. So let's focus on this rectangle on the left first, the baseline metabolism. So every living organism has, you know, this baseline metabolism it has to fund, right? So even if you spend all day on the couch watching TV and you have somebody bringing you food and doing everything for you, your body is still using energy, right? It still has to keep all your cells alive, and whatever minimal amounts of activity are going on in your brain have to be funded through that baseline metabolism. Plants have the same thing. In order just for those plants to stay alive, they have to have a baseline metabolism. So that kind of is a priority for plants. Plants are taking a certain amount of carbon and they have to fund that baseline metabolism. But after that's funded, if you will, or that's covered, then it can quote unquote choose. Plants aren't choosing, right? But they're triggered because of different stimuli to allocate carbon to these other different categories. So it could produce new leaves. It might produce stem if it needs to grow taller. It might produce roots if it needs to access below ground resources. It might decide to reproduce. It might be in a harsh environment with lots of herbivores and it has to produce defenses. Or it might say like, I need to store carbon for growth next year. And so it could put carbon towards storage. So these are the different types of categories a plant might allocate carbon to. What I really want to drive home here, though, is that you've got these different categories, but there are trade-offs associated with how plants allocate carbon. So if a particular plant, let's go to a couple examples here. So this drawing here, I haven't rearranged the text, I just squashed boxes and enlarged boxes. But this represents a plant that might allocate most of its carbon going to growth to leaves. And so the leaf box here got larger compared to the last slide, but that came at a cost of stem growth and root growth. And so if a plant allocates lots of carbon to one thing, that reduces how much carbon it has for those other categories. Similarly, let's say a plant is exposed to lots of herbivores, and so it produces lots of defensive compounds. And that takes a lot of energy to produce those compounds, and that means it would have less carbon to allocate to these other categories. So here I've drawn storage and reproduction really small, but instead it might still allocate lots to reproduction and just not allocate much to new growth. So there's lots of different combinations of how plants could allocate carbon and reduce carbon to other categories. And how plants allocate their carbon has a lot to do with what type of environment they're best adapted to. All right, so that's kind of why we use carbon budgets and thinking about carbon moving to different parts of the plant to understand plant growth strategies and plant success. Okay, so I'm going to take a short break because talking this long at once, I'm going to start fumbling my words. So I'll take a little break. Come back in a second and we'll start talking more about that second step of photosynthesis, actually getting CO2 or harnessing and fixing CO2. Okay, I'm back, and now let's talk about those CO2 reactions. So we're going to talk about a particular enzyme a lot, and that's rubisco, which is the world's most abundant enzyme. So this enzyme is in every plant or organism that does photosynthesis. And so if you think about how many plants there are and how many leaves there are, you start in the world, you can start to get a feel for why this might be the most abundant enzyme on Earth. First, I want to talk about the name rubisco. So rubisco is like the abbreviation. The full name is ribulose bisphosphate. It actually is ribulose 1,5-bisphosphate carboxylase oxygenase. But we shorten it to rubisco. But you need to be familiar with that name, and that's mainly because this carboxylase oxygenase, kind of the reactions that it can do are going to be really important. But for now, we'll just call it rubisco. This is just a picture of the structure of rubisco, which isn't super important to us. But if you haven't had biochemistry yet, or maybe you never will take it, you should know that enzymes, there's this very particular structure to enzymes such that whatever molecule an enzyme is going to react with, that molecule fits into a little port, if you will, or a place on that enzyme. And so that structure of the enzyme is really important in the reaction it speeds up, or it helps with. So this is just the general reaction of photosynthesis. You've probably seen something like this before, but when a plant does photosynthesis, every time it fixes or combines 6 molecules of CO2 with 6 molecules of water, it can produce this particular simple sugar here and then release 6 molecules of oxygen. And again, this whole reaction isn't performed by rubisco, but rubisco performs that first and primary step where it's fixing that CO2. So it's the most, not the most important, it's the most abundant it's the most abundant protein on Earth, or enzyme on Earth. Enzymes are a special type of protein. The other thing I want to point out is that nitrogen is a really important element of all enzymes in plants, but especially in rubisco. And so if a plant is nitrogen limited, there's a good chance it's not able to produce as much rubisco as it might want to, and that would affect its ability to take up CO2 quickly. Because the more rubisco molecules that are in the leaf, the faster it can take up CO2. So it doesn't have just one rubisco molecule in the leaf, it's got tons of rubisco, and all of those rubisco enzymes are taking up CO2. So the more rubisco in a leaf, typically the faster the rate of photosynthesis is going to be, because it can take up that CO2 faster. Okay, one other thing about photosynthesis and this particular step we're talking about is that for the vast majority of plants, over 90%, it's a little over 90% of plants use a particular type of photosynthesis where the first product of photosynthesis is this molecule right here. It's 3-phosphoglycerate. Now, you don't have to remember the name of this product. What I want you to remember is that this first product has 3 carbons. The red C's are representing the carbons in that molecule. Because the first product has 3 carbons, we refer to this type of photosynthesis as C3 photosynthesis. Plants, we're going to talk about 3 different types of photosynthesis. Each species can only utilize one type of photosynthesis for the most part. There's a couple of species that can do this funky thing and switch back and forth. But for the most part, a plant uses one type of photosynthesis. And so if a plant utilizes C3 photosynthesis, we often call it a C3 plant. All right, so that's kind of the language we're going to use. Again, here's just great. More than 90% of plants utilize C3 photosynthesis. So that's why we're going to start talking about it. And then we're going to talk about the different types of photosynthesis and contrast them to C3. Okay, let's talk about, let's just skip this slide for now. We'll come back to it. Okay, so there are trade-offs associated with photosynthesis and doing photosynthesis fast or quickly. And so that's what we're going to talk about now. This figure shows that fundamental trade-off. You're all familiar with it, but I just want to drive home how fundamental this is to plant growth. And it's really at the heart of kind of determining the distribution of plants around this globe and how plants can deal with this trade-off. So this figure here shows the rate of stomatal conductance to water, which is on the x-axis. So this is basically telling us how fast water is leaving a leaf. So the further to the right on this x-axis you go, the faster a plant is using water. On the y-axis here we have A, or assimilation, which is the same as photosynthetic rate. We just use a capital A to abbreviate assimilation. The further up on the y-axis you go, the faster a plant is assimilating or fixing carbon. What this graph shows is just that tight correlation between how fast a plant can fix carbon and how much water it's using. So plants that are high up on the y-axis, meaning they're doing photosynthesis really fast, are also on the far right of the axis. That means they're using lots of water. And this is actually a single species. And these plants at the right were under well-watered conditions, and as the soil dried out, I continued to measure these 2 variables. And so these plants down here are representing plants that are drought stressed. And so what's happening is, as the plants are getting drought stressed, they're reducing how much water they're using, but that's coming at a cost of doing photosynthesis. And although this is for a single species, if we were plotting different species along here, it would look much the same. So just to give you an idea of that trade-off between fixing carbon and losing water, a lot more water molecules are lost for every molecule of CO2 fixed. And approximately 400 molecules are lost for every 1 molecule of CO2. So if a plant is doing photosynthesis, it's coming at a huge cost to water use. Okay, so now let's talk about how that CO2 is getting into the leaf and where this trade-off comes from. So we're going to go back to this picture here. So this picture depicts two different stomates on the surface of a leaf. Now, on the surface of a leaf, there are thousands and thousands of these stomates. They're actually made up of two cells, which you can't really tell in this picture, but one cell would be on the upper portion here, and it would be touching a second cell on the lower portion. And these two cells can come together and seal off, or they can move apart and open up like this stomate that's shown more on the bottom right part of this picture. So when these two cells are open, you've got this open pore where CO2 can diffuse into the leaf, but this pore can't discriminate against different gases. And so when it's open, CO2 may be diffusing in, but water vapor and other gases might be moving out. Similarly, when it's open, we've got CO2 coming in, but we've got really just atmospheric gases moving in. So we've got oxygen and nitrogen and other compounds in our atmosphere that are diffusing in at the same time. So that's where that trade-off comes in, because if it's open, you're going to get your CO2, but you're going to lose water all at the same time. Let me see, do I want to skip? No, I think I'll go ahead and describe this image to you a little bit so we can see where things are happening. So this is a not as high quality image as what we looked at before, but this is a cross section of a leaf, and this is a grass leaf, and it is a C3 plant. But this structure that I'm going to talk about applies to all C3 species. So up here in this upper part of the picture, it's again not as clean as the other one, but this is the stomate right here. And so there's this cell on the right and there's another cell on the left that can open and close. Inside the leaf, just adjacent to that stomate, is an air space we call stomatal cavity. Then we have some cells here, and this black stuff in the cells, a lot of this is the rubisco. So the rubisco in C3 plants is in these cells out here, so that when CO2 comes into the leaf, the rubisco is right there so it can grab those CO2 molecules, fix them, and continue on with photosynthesis. This structure here is again that vascular bundle we talked about. These are the bundle sheath cells. You can see that ring of cells around the bundle sheath. These three large pinkish cells are the xylem cells moving water. And then this group of cells up here is the phloem moving carbohydrates and nutrients and hormones and other compounds. All right, so that's kind of where things are happening and where the rubisco is relative to that stomate. The other thing to keep in mind is that these cells are all fully hydrated when a plant is alive and working. And so when these stomates are open, you can see there's a really quick path for water to convert from liquid in the cells to vapor and diffuse out of there. And the atmospheric air is, except for when it's raining, atmospheric air is relatively dry compared to the cells in the plant. So that water is definitely leaving those cells whenever the stomate is open. Okay, so just to review, we broke photosynthesis down into 3 steps. We talked about rubisco, and remember, you need to be familiar with the name of rubisco, and why that is will become clear here in just a minute. Remember what the first product of photosynthesis is in 90% of plants, in C3 plants, and how CO2 is entering the leaf and what that trade-off is. But we've talked about several things related to the movement of CO2 into the leaf and the water vapor lost and those steps of photosynthesis. And so based on what we've talked about so far, we can start thinking about what is going to control the rate of photosynthesis in different plants and in different locations. And so step 1 is harnessing solar energy. So in order to do photosynthesis at a fast rate, you have to have lots of solar energy. So that's criterion number 1. Criterion number 2 is that you have to have lots of CO2, right? In order for rubisco to be fixing that CO2, you need a steady supply of CO2 into the leaf, which requires that open stomate. But in order to keep that stomate open, we know a plant has to have access to lots of water because water is going to be lost all the time. So you need lots of CO2, you need lots of water. And then lastly, we talked about rubisco being, or not being made solely of, but needing nitrogen to make rubisco. And so if a plant doesn't have access to nitrogen, it's not going to make a lot of rubisco, and it's not going to be able to fix CO2 as fast, even if the stomates are wide open. So it needs all of those things in order to do photosynthesis really fast. All right, and that's primarily thinking about a C3 plant. Okay, we're going to talk a little bit more about C3 before we contrast it to the other types of photosynthesis, but let's just talk about what those three types are. So C3 is the most common type, and remember, just a little more than 90% of plants utilize C3. C4 is the most, is kind of the intermediately used. It's not really a phrase, but anyway, it's kind of the intermediate type of photosynthesis we see in plants. And then CAM is the least commonly used type of photosynthesis. All right, so let's go back here and think about rubisco a little bit, and let's think more about these last two letters in the name of rubisco. Remember, rubisco stands for ribulose bisphosphate carboxylase, which is the C, oxygenase, which is the O. And so that means it can have two types of reaction, or it performs two types of reactions. So we'll talk about the carboxylation reaction first. When rubisco reacts with a CO2 molecule, so remember, an enzyme has this structure and a molecule can fit into it. So when a CO2 molecule fits into that rubisco, that's a carboxylation reaction, and that's a net gain of carbon for the plant, and so that results in positive plant growth. I'm going to move these words here. They kind of got out of their original location. So we can think of carboxylation as being good for a plant. It's gonna get carbon, it's gonna build its pool of carbon up when it can do the carboxylation reaction. Turns out though, rubisco, like other molecules, well, one other molecule can fit in that reaction center of the enzyme. And so oxygen is the other molecule that rubisco can react with. Unfortunately, when an O2 molecule gets into that enzyme and reacts with it, we end up with a net loss of carbon from the plant. It actually ends up respiring a CO2 molecule back out. Remember, it takes a lot of energy to get carbon in, and so if it's doing a reaction that loses carbon, that's a bad thing for a plant and results in reduced plant growth. So plants want to minimize how much oxygenation, or how many of those oxygenation reactions are occurring. The word we use for oxygenation reactions is photorespiration, because it's releasing a CO2 molecule, it's respiring during photosynthesis. So photorespiration is a bad thing for plants, and it reduces plant growth. So again, plants want to minimize how much photorespiration is occurring. All right, let's talk about a C3 plant again. So this is a different image of a C3 plant. You can see the stomates a little bit better here. So when the stomates are open, CO2 is coming in. But remember, all of the other gases in the atmosphere are also coming in, including oxygen. If you, our atmosphere is about 20% oxygen, and it's about 0.03% CO2. So it's a much smaller amount of CO2 in the atmosphere than it is. I'm trying to check my calculations. Let's just say CO2 is a lot less than, I forget where the decimal place is. Well, maybe we can talk about it in lab. Much less than 1% of our atmosphere is CO2. A lot of it is oxygen. So when the atmospheric gases get into this stomatal cavity, there's a lot more oxygen around than CO2. Now, rubisco reacts more readily. It fits with CO2 better. So it's going to react with CO2 more often than it does oxygen. But it photorespires a lot just because there's so much oxygen there. The other thing to think about is if a plant has to close its stomates, what's going to happen inside this stomatal cavity is that rubisco is going to react with all of the CO2 molecules that it can, and then there will be no more CO2 because we don't have the stomates open, we don't have a new supply of air. Then it will start reacting with those oxygen molecules because it can, and it fits with them, and it will start reacting with those. So in order for a C3 plant to minimize photorespiration, it needs to keep its stomates wide open so a new supply of CO2 can continually flood that stomatal cavity. And you know that means lots of water is coming out. So if we think about C3 photosynthesis, we know, and this isn't just in C3 plants, but rubisco can react with either CO2 or O2 in order to maximize carboxylation and minimize photorespiration. Plants have to have high stomatal conductance, or those stomates need to be wide open, which means it's using lots of water. Turns out there's also an effect of temperature on rubisco and its ability, or its kind of affinity for CO2 or O2. This graph here shows leaf temperature on the x-axis in Celsius. So high leaf temperature on the right, low leaf temperature on the left. And this symbol here is the rate of photorespiration. So high photorespiration, remember that's a bad thing for plants, and low photorespiration lower down on the y-axis. You can see that as temperatures of the leaves increase, the rate of photorespiration goes up. And that's because the structure of rubisco changes a little bit and it starts fitting better and better with oxygen. And so it starts reacting more and more readily with oxygen. So what that means, okay, let's jump down here. I've got a couple slides in there I probably shouldn't have, but that's okay. Let's jump down to this slide. This is, oh no. All right, let's not do that. Let's go up here and actually I'm gonna add some text here. And it's probably gonna mess us all up, but that's all right. So what that means for a C3 plant and its habitat is that it needs cooler conditions, cooler temperatures, and it needs lots of water, right? So it needs to be able to keep its stomates open. It means it needs access to lots of water, and it does better in cooler temperatures so that that rubisco doesn't change structure and start reacting with oxygen a lot. All right, so now let's move on to C4 photosynthesis. So this is the structure of a C4 leaf. This structure here is a stomate. It's a little harder to see. And then we've got cells near that stomatal cavity. And then if we look at our vascular bundle, it may be not clear right now, but if you flip back and forth between the images I've shown you, the cells in this bundle sheath are much larger in a C4 plant than a C3 plant. And that's because all of the photosynthetic machinery in a C4 plant are in this bundle sheath. Remember, in a C3 plant, the rubisco and the other pigments that are part of photosynthesis, they're in these cells out here. But in a C4 plant, much of that machinery is in the bundle sheath cells. And then remember, we've got the vascular bundle in here where we've got water and carbohydrates being moved around. All right, so this is important because now rubisco isn't directly in contact with the CO2 and the oxygen that come in through the stomate. Now for getting carbon, that's a bit of a problem, right? Because if it's not close to the CO2, how's it gonna fix it? It's good for photorespiration because it doesn't have access to the oxygen, and so C4 plants don't photorespire a lot. So in order to get that CO2 and the rubisco together, C4 plants utilize a new enzyme, or I shouldn't say new, a different enzyme. All plants have this enzyme called pep carboxylase. Turns out C4 plants just use it a little bit differently. So in a C4 plant, I'm going to go here again, pep carboxylase is out here in these cells. It reacts with carbon first and then moves that carbon into the bundle sheath cells and hands it off to rubisco, where rubisco grabs it and then can continue on with photosynthesis. The first product of photosynthesis in a C4 plant is, as you might guess, has 4 carbon atoms in it. So it's a 4-carbon compound, and that's where we get the name C4 photosynthesis. C4 plants also have this specialized anatomy, which is these enlarged bundle sheath cells, and that's referred to as Krantz anatomy. Because rubisco doesn't have access to oxygen, it means that C4 plants can have a lower stomatal conductance, or they can keep their stomates closed a little bit. Remember, C3 plants have to have their stomates wide open in order to get a new supply of CO2 so that Rubisco can always find new CO2 molecules. But since PEP carboxylase is the first enzyme, and it's only a carboxylase, it's not an oxygenase, it doesn't need to have near as many carbon molecules around in order to do photosynthesis. That allows that allows C4 plants to have lower stomatal conductance. Because our oxygen is separated from Rubisco, so oxygen is out here by the stomate and Rubisco is in here at the bundle sheath, they're spatially separated. That allows these plants to also tolerate higher temperatures. Even though Rubisco still changes its structure a bit under warmer temperatures in C4 plants, there's no oxygen around to react with, so it doesn't really have a big impact on C4 plants. So here is the figure I showed you before that shows the relationship between leaf temperature on the x-axis and photorespiration on the y-axis. Remember, this line here is our C3 plant, and these lines down here are 3 different C4 plants. You can see the rate of photorespiration is much lower. It still increases with temperature, but it's still way lower than a C3 plant. And that's, again, because of that spatial separation between Rubisco and oxygen. So these are take-home messages. C4 plants have higher water use efficiency, or they can conserve water compared to C3 plants. They can maintain growth, positive growth under higher temperatures. And so their habitat is more likely to be, or they do well under warmer and drier conditions than a C3 plant does. They still do great under well-watered conditions in cooler temperatures, but there is a trade-off associated with C4 photosynthesis. Because there's a whole extra step and there's some movement of Pep-C from one place to another to move that carbon to where Rubisco is, that takes extra energy. And so if you're going to put a C4 plant right next to a C3 plant in a cool and wet environment, that C4 plant is using more energy to do photosynthesis than C3 plants. And it doesn't need to do that extra energy step in that cool and wet environment. And so that C3 plant is going to outcompete a C4 plant most of the time. But if you were to move those two plants to a dry environment, now all of a sudden it's an advantage to do that extra step. So even though it takes more energy to do photosynthesis, it can keep photosynthesizing at those hotter temperatures and drier conditions. And so in those situations, that C4 plant is gonna outcompete the C3 plant. Okay, so that's our trade-off. Now I'm gonna take another little break because you can tell I'm starting to mix up my words more than normal. And then we'll come back and we'll finish up by talking about CAM photosynthesis. All right, now let's talk about CAM photosynthesis. I have a couple of pictures down here of different types of cactus or succulent plants, and that's where we find CAM photosynthesis most often utilized. There are plants that are not cactus or succulents that utilize this type of photosynthesis, but that's where it's most commonly found. Okay, so just reviewing C3 photosynthesis again, remember that its first step produces a 3-carbon molecule. And in order to minimize photorespiration, it needs to open its stomates wide, which means it uses lots of water, and it needs to be in relatively cool temperatures so that when Rubisco changes its shape a little bit and more readily reacts with oxygen, Rubisco's not, well, that happens when it warms up, and so it wants to minimize that effect in C3 plants. So what I'm trying to say is remember that C3 plants typically do best in cooler and wetter habitats. So now we talked about C4 photosynthesis. Now there's another type of photosynthesis that can minimize photorespiration a different way. And so CAM stands for crassulacean acid metabolism. Crassulaceae is a family, a plant family, and that's where it was first identified, and so that's why it's referred to as crassulacean acid metabolism. Again, it doesn't only happen in the family of Crassulaceae, but that's certainly where it's most common and where it gets its name. So in in CAM plants, or in CAM photosynthesis, the first step in photosynthesis is also PEP carboxylase. And that's because when CAM plants photosynthesize and first fix CO2, they actually do it at night. It turns out Rubisco isn't active at night. Rubisco is triggered to be active due to solar energy. And so when there's no solar energy coming into a leaf, Rubisco is kind of like dormant. It goes quiet and it waits for that solar energy to come. So in order for CAM plants to photosynthesize at night, they have to use a different enzyme, and so they utilize PEP carboxylase. The other interesting thing about CAM plants is because they're photosynthesizing at night, they're opening their stomates at night as well in order to get CO2 into the leaf so PEP carboxylase can have access to it. That's also really interesting because for C3 and C4 plants and the way stomates evolved, they evolved to open in the daytime. Like their main trigger for opening is sunlight. And so CAM plants basically had to reverse that so that it could open its stomates at night instead of the daytime. So it's a really cool like evolutionary step to make such a major switch. But what CAM plants do is they open their stomates at night so they can get CO2. PEP carboxylase reacts with CO2 first and then stores it in this vacuole. So a vacuole of a cell is just like a big storage area. And so that's what happens at night. Then during the day, the CAM plants close their stomates, which allows them to conserve water. Rubisco is activated during the day, and so they pull that carbon back out of the vacuole and give it to Rubisco to finish photosynthesis. By opening stomates at nighttime, relative humidity goes up, the cooler temperatures minimize how much transpiration occurs in a CAM plant. Then again, it can still harness the energy from the sun during the daytime. And I don't have a nice diagram or anatomy of CAM plants because they're all different. It's not a uniform kind of structure because Rubisco isn't spatially separated from oxygen in a CAM plant. It's temporally separated. So I keep stressing the words spatial and temporal. So you'll use those words and descriptors of C4 photosynthesis and CAM photosynthesis, right? So just a summary of CAM. Remember this photosynthetic pathway or mechanism is named after the family Crassulaceae, which is where it was first discovered, but remember it's not restricted to that family. And it's got a temporal separation between Rubisco and oxygen. It's really high water conservation. By opening at night, it really is able to conserve a lot of water. But there is a bigger trade-off with CAM plants than C4 plants. They're really slow growing because CAM photosynthesis takes even more energy than C4 photosynthesis. And so the only plants that grow fast enough to dominate an ecosystem are going to be growing in really, really hot and really, really dry conditions, even more so than C4 photosynthesis. So plants that utilize CAM photosynthesis are well adapted to really hot, really dry conditions. That's why lots of cactus and succulent utilize CAM, and that's why they're dominating our desert ecosystems because they're so hot and so dry. And that's the only type of photosynthesis. Well, it's not the only type of photosynthesis that works there, but it's the best adapted type of photosynthesis in those really harsh environments. Okay, so those are 3 types of photosynthesis. There's a worksheet that you'll need to complete associated with this lecture. And so I'm going to have you complete that for this week, but then we're going to go over all of those questions in a couple weeks to make sure you feel really confident in your answers and these mechanisms before our exam in week 6.